come to your talk.
Okay, thanks. Afternoon, everybody. Thanks to Gina and Chris for inviting me to what is a fantastic symposium. I'm really delighted to be here. I was to be a co-chair with Alan on part of the malaria series that Bruno alluded to earlier, and we'd had a really lively conversation about the concepts of what would comprise that document, and I have to say it was made much harder by his passing, and I'm sure he would have made an excellent job of it. It should be out soon, and so you'll see the fruits of that work. So I'm going to follow on from Diane really and talk about how we can use how our body reacts to that diverse array of parasites it's exposed to look at malaria transmission dynamics to increase our chances of identifying where transmission is occurring, to better target control and elimination activities. And the unique facet of antibodies is that they last for much longer than the infection in the mosquito or the human. And so what I'm going to try and do, the outline, is just to try and take you through why serology might be useful, and then to go through some of the developments that we've had recently, and to highlight the challenges of an adoption of any new technology. So we're going to start, why is it useful? So as I said, the infection in the mosquito is a matter of potentially days or not very much longer than weeks. In humans, I think there's some debate about how long infections will last depending on the level of immunity, but it's certainly not as long as the immune response that infection will generate in the human host. And we've had some various estimates of that from years to, I'm not getting the hang of that. Various estimates of that from 5 to 40 years, depending. You can look at individuals who've migrated to non-endemic countries and still find antibody responses. What you will see is that the levels of antibody responses drop over time, and that becomes relevant, and I'll talk a little bit more about that. So practically, what does that allow you to do? So in that schema here, what I've got are a series of parasite surveys in this. This is a small population, but you can scale it as much as you like, and they can be as frequent as you like. I'm talking about potentially surveys every 2 months, every half year. And this is what we see with our parasite infections. We can see that 3 of those surveys might be negative. If we were looking for malaria parasites, we might assume that there was no infection in that population. But if we did a serological survey, what we would expect is to see the footprints of that infection within that population here, and with some nod to the amount of detection we would have since the time since that infection originally took place. So that infection was that long ago, would have the lowest level of antibodies. So as its very simplest, what this approach allows is the detection of many more infection events or exposure events than simply a single time point parasitological survey. So when we're looking for these increasingly rare, sporadic infections, the idea of having a more sensitive diagnostic that would be an additional tool, was not proposing it to be the only tool, but as a tool in your toolbox, to be able to use that to detect exposure events is really potentially very useful. And so what would that look like if I now try and look that over time? This is some work that we've done with Edward Wenger at IDM in Seattle, thinking about how we might perceive the use of a serological test in an endemic population. And what you can see, probably just make out in the bottom here, is what we have is our RDT with relatively low detection threshold, PCR with a little bit more, but then we're looking at antibody levels associated with different time periods of antibodies with different half-lives, essentially, with a longer half-life of, say, something like 18 months in the light green here. So already you can begin to see how much more transmission or exposure we would detect by looking at by incorporating some sort of antibody into our survey methodology. as there has been quite a lot of, the way that we interpret serological data here is simply we can either look at a seroprevalence measure, so the proportion or percentage of individuals that have antibodies, or typically as exposure to the parasite is related to age, we can look at fitting a simple line through that age seroprevalence, which gives us a seroconversion rate, i.e. that's the rate of individuals which become antibody positive over the course of typically a year. And what we've shown is that essentially that seroconversion rate, which is the blue triangles there, is much more tightly correlated with the entomological inoculation rate than the paired parasite rate derived from those same samples. So it's a much more consistent measure of transmission than parasite rate. As I showed you in the previous slide, parasite rates can vary depending on when you go. Seasonality could be just after drug treatment. So there's good provenance there for this as a method. And then just trying to give you a sense of the scale, I borrowed this from another one of the Malera outputs on interventions and modelling, looking at the range of transmission which we might expect and the other different markers or measures of transmission that we might commonly encounter, such as EIR, parasite rate, weekly cases and annual parasite index. And again, these are predicted seroprevalence and seroconversion rates. The thing that is evident here is that at this very, very low transmission level where we are finding it difficult to measure and parameterize the conventional measures, serological measures are actually giving us quite a clear indication, or we can begin to still grade transmission intensity within this very low range. But it's not new. It's been done before. There's a nod to the history that we heard from Pedro and Bruno. Here are two sort of classic examples of using seroprevalence before. This is Mauritius, the elimination on Mauritius in 1973 by Bruce Schwott. A lot of students will have read his classic textbook. And this is looking at seroprevalence 5 years after eradication on Mauritius, or elimination, I should say. And what they then there is showing, as you would expect, in children born since transmission has ceased, no evidence of exposure, no antibodies. So what we have here is this unique facet of malaria, of serology, in the sense that it recreates the exposure history over time, and we can begin to look at changes in transmission intensity. And here what he did rather simply, probably just as good as some of the models that we've done, is just simply fit regression models to get an idea of how the transmission intensity at different age groups and different parts of time. Here is the classic Garkey study, looking at this kind of filled area is the antibody levels, so these are levels rather than prevalence, in the control groups, and the lines going down are in the several weeks following intervention. So what you can see is population antibody levels decreasing over time, and then when they stopped the intervention, population antibody levels returning back up to pre-intervention levels as transmission is re-established. So there's a strong historical precedence, and when I first started to talk to Alan about serology, he said, well, why don't you just do that? They've done it before and it works. And so he was supportive, but not necessarily effusive about how modern technology might adapt to this. And so his first suggestion was that we try to see whether commercial kits that are available for screening blood transfusion, certainly we use them in the UK and other European countries use those, to see whether they could be adapted for epidemiological purposes, and we're in the process of doing that, but whilst it's a very important piece of work, it's actually quite boring, so I'm not going to show you any of that. But he was always challenging us to say, well, okay, if this isn't good, and Diane gave a very good kind of idea as to why it might be the variation in parasite development, and also our inherent human genetic variation as varying responses, then what would be the new platforms that might take this forward? So I've collated some information from various people who are working in the area now, just to show you kind of how we're moving forward with the various approaches to generating serological data. So the whole idea of the previous historical work, what they used was essentially homogenized parasites. And what you have in a homogenized parasite, obviously, all the antigens that the parasite expresses, and not only that, but they are in the right relative amounts to each other. However, they're typically only one strain of parasite, and it's not so easy to produce vast quantities of those antigens in a standardized way. So some work kind of initiated and led by Phil Felger at UC Irving began to look at, well, let's look at all the malaria parasite proteins and see how serology can be used to identify and to hone down important antigens that might be used for, obviously serological markers of exposure, but also various other approaches such as vaccine development. So what Phil did was essentially look at all 7,000 odd proteins of malaria parasites and examine antibody responses to all of those. And what you can see is that actually relatively small proportion of those actually are responded to by the human sera. So that's actually quite good because it allows us to hone down on immunogenic antigens, ones that might be used as serological assays. You can see here his panel of naive US adults and then some naturally exposed or some sera from naturally exposed Africans here, lighting up his microarray panel. So what we've got are at least a reduced range, but still a range of antigenic targets that we might incorporate. And Phil and his colleagues at UC Irving, together with many investigators in the NIH-funded ICEMA program, are now beginning to examine antibody responses to this kind of honed down list of antigens as part of a multi-site survey. Here he's shared some data with us from the 3 different areas of Zambia, looking at differential responses to the top 30 antigens between children and adults in varying areas of varying transmission levels. And you can quite clearly see that not only does the response scale with the amount of malaria there is, but it's also lower in children and adults, suggesting that it's on the right track. Chris's group have taken that further. So as Diane described, the highly variant antigens, multiple different clones expressing variant antigens, or antigenic variants in this case, of some key Merozoite stage antigens. So Chris, together with Phil, has developed a panel or a microarray of variant AMA and variant MSP1 antigens. And the relatively simple take-home there is that adults react to more variants, i.e. they see more clones in their lifetime than children, but the number of variants that children recognise increases after the transmission season, which is again something that you would expect to see. But the relevance there is that actually we can begin to potentially really finely dissect the response to not only have an underlying molecular kind of characterization of the parasites, but use that to overlap with exposure to those parasites in the population. So Chris's group have taken this even further and drilled down a lot more. So the previous array was based on proteins. Here what he's done is an array based on overlapping peptides. Some of us will remember the old peptide array PEP scans that we used to do on ELISA plates, very time consuming. Now the microarray technology has completely revolutionized our ability to do that, to look at that in depth and in large numbers. But the novel element here is that what is also on the array are peptides from Anopheles saliva antigens. So obviously when a mosquito bites, it inoculates saliva as an anticoagulant, and you as an individual will develop an immune and antibody response to that saliva. So potential marker for exposure to bites. So again, this is a really neat way of drilling down onto identifying markers of exposure to mosquito bites. And the panels show antigens from different Anopheles species and their relative seroreactivity in North American adults, Southeast Asian adults, and Malians. And the Southeast Asians are clinical cases, I believe. How we then use those in an epidemiological context is potentially where we're really interested. So whilst we have the technological laboratory-based development, what we have is a desire to deploy and test those tools epidemiologically. And this is data from Vincent Corbel and work on the Thai-Myanmar border, showing essentially heat maps of high antibody level responses to Anopheles proteins, correlating with the red dots, which are high levels of anopheline catches and EIR. So again, the potential to use that as predictive to where the bites might be happening and to target your interventions accordingly. So I talked a little bit about the beginning about the identification of antigens of recent exposure. One of the issues with serology is that, or antibody responses, it does last for a long time. What we've been interested in, together with colleagues at UCSF, is identifying individuals or identifying antigens that are associated with short-term antibody responses. So in this bottom left panel here, what we have are a single individual, time since exposure, all the antigens there measured against, or that's one antigen against a series of individuals, looking at the decline since the time from infection. And there we're identifying all the antigens which show that same profile, a decrease since the time since exposure. So when we begin to try and predict time since exposure or incidence of infection, if we use 600 antigens, that prediction is not very tight. But if we use the top 10 antigens from this particular plot here, we can find that this is time since exposure, so a rapid reduction in antibody levels. This is incidence, an increase in responses to those antigens. So what we've done in London is make those antigens. We've actually produced them and we've put them onto the Luminex platform and the bead array platform to look at kinetics post-exposure in a very detailed cohort that our colleagues at IDRC have been managing together with UCSF. And here are individual plots, and you can probably just make out the key at the bottom there, a malaria infection, and what we can see are select antigens dropping off since the time since exposure. And when we combine those antigenic responses, what we can see here are some of the top 10 hits of the ones we've managed to actually produce recombinantly, dropping quite rapidly after time since they were infected, whereas some of the more classic antigens show a much shallower drop in antibody level. So Pedro talked about Plasmodium knowlesi, and I think that we're kind of interested in understanding the response to other species because we want to have a fidelity around what we understand our response to falciparum. And also in terms of knowlesi, it's a relatively rare event, and it allows us a kind of a biological attempt to look for rare events within a population. So what we've done here is the opposite. We've tried to identify antigens which have no sequence homology with Plasmodium vivax, for example, because there's quite a lot of sequence homology, but also antigens which exhibit heightened immunogenicity, i.e. are likely to induce an antibody response. So that's based on hydrophobicity, it's based on whether they're membrane bound, etc. So a series of qualities that define immunogenicity. We then produce those in a recombinant way and put them into ELISA in a population survey from Malaysia. This is our falciparum plot, which shows very, very little falciparum exposure. As Pedro said, Malaysia is quite clearly on the way to elimination, but there was some historical exposure. This is the knowlesi plot, nothing like a pattern that we could observe, suggesting that transmission is very, very different, and that's what we know about Plasmodium knowlesi. The risk factors are not specifically age-based, but they are spatially. So when we put those on a map, this is that particular plot. This is Borneo, Malaysian Borneo, and this is the district of Kudat. What we can see for Plasmodium knowlesi is that the antibody responses tend to be on the outside of the village, in closer proximity to forest, and the falciparum responses that exist tend to be peridomestic. So also advances, how do we use this in terms of an analytical sense? And this is just a quick flash through some methodologies that allow a sample size to be developed if you wanted to use the assay as Alan had originally suggested it for elimination, certification of elimination. And here, a different approach for freedom from infection, where we're thinking about actually the probability that disease does not exist. And Gillian Stressman has a talk on this. This is something that we've been developing. So finally, one of the main things that we can do with the serological approach, simply because you do get many more seropositives, is develop a mapping approach. And this is some recent, definitely hot off the press data from Myanmar, the recent survey conducted by Malaria Consortium, DMR, in Myanmar. This is the original Malaria Atlas Project map, which is not particularly informative, or primarily based on the lack of information going into it. This is the annual parasite index map that is used to define the surveying structure, the cluster survey. And this is the serological map that has been developed with collaborators Pete Gething and Ewan Cameron in Oxford. And what you can see immediately is a lot more granularity. It maps it traces the API very well, but it also provides a lot more granularity in the mapping. So that's something that I think offers real possibilities. So the challenges, I've got one slide, but this is really where most of the endeavours need to go. What I've described is a variety of platforms providing a massive amount of data. And our challenge is to translate those multiple antigen responses across the top from multiple people into a series of, well, yes or no, is that positive, is that not? And then to say, well, okay, we've got multiple antigens, what do those antigenic responses mean in terms of historical exposure or recent exposure? And then actually, how does that fit when this population come from one particular area? How am I going to decide what that antigenic response means, the seroprevalence here, for these particular areas? And how does that translate to a village layout or a region layout in order to specifically guide some sort of control or intervention procedure? And that's really where the challenge is at the moment. And I remember discussing this with Alan some months before he passed away. And he was saying, well, you know, it looks very promising, but it is slightly aspirational at the moment. And I kind of reminded him that a little bit of aspiration wasn't a bad thing. So I think that my summary would be that the antibodies has an excellent potential as additional measure of transmission, particularly in low transmission settings, with that unique facet of being able to look back in time, which none of the other metrics that we have are able to do. But the major issue is providing a standardized assay with clear use scenarios. And then I'd just like to thank a few people that have provided data and helped along the way. So thank you.